Thank you very much. Thank you, everybody, for joining. And as Bijan said, I'm going to talk about our latest model of our 64925 LC triple quad, always affectionately known as the D model, and why this is such a good instrument. It's for looking at omics. It's a multi-omics platform, and for standardizing these big, complicated, challenging sort of metabolomics, in this case, study. So those of you that are familiar with us, you will know that we like to cover the entire omics workflow, right from the sample prep through the LCMS side of things and data analysis, and also the reporting. So just to put some context into my talk, before talking about the triple quad, why it's so good to use these standardized methods for these metabolomics and lipidomics studies. So a traditional discovery workflow would typically use a high-res instrument, and in our case, it would be a QTOF. You would go through the feature extraction, so you have to pull out all of these unknown features and match them and align them across all of your different samples, and this can be quite laborious. Then you can do your nice statistics, and you can find your significant metabolites, which is lovely, but then you've got the whole process of trying to identify them. Then you can build your routine MRM-based targeted assay. So what we want you today to think about is to actually think about, can I actually do some discovery work using a triple quadrupole? I've got a standardized targeted method, I've got a large cohort or panel of metabolites, and can I get meaningful, statistically relevant data out of this? And hopefully by the end of this talk and by the end of seeing Manuel's talk, you'll be convinced. So our workflow would then look something like this. In this data acquisition, we'd have tens to hundreds of metabolites. We can then extract all of that data and perform our chemometrics, because chemometrics is all about extracting information from chemical systems, and it's all data-driven. From that, we can then actually discover something new. So we know what our metabolites are, we know what pathways they're involved in, and we can discover how they've been impacted by our samples and by our experiments. We can then identify those now significant or interesting metabolites and build a less complicated routine assay until we then build our next experiment and go through the process again. But our methodology will, on the LCMS side, will always be the same, and it makes it very fast and very standard. So in order to do that, we need a robust, reproducible and routine and an instrument, and be able to perform everything in a routine way. And these got some key features on our 95D. So the first thing is the tuning and the calibration. And actually we use artificial intelligence in order to perform what's called swarm tuning. And this is a really clever algorithm, because it means that multiple parameters can be changed. And so it speeds up the whole tuning and calibration process. We have active system monitoring. So there's early maintenance feedback. And so this alerts us to when we need to perform maintenance events. And it means that we can anticipate these coming up rather than wait for something to either have run out of its time period and it needs changing, or you have a problem on your sample. So we can automate that maintenance as well. So one of the nice things now is that you can actually schedule your auto-tune or your check tune. So you can set it to come on in the morning so that by the time you come into the lab, it's all done, it's set up and ready to go. We also have a fully automated MRM and ion source optimizer. And so, of course, this really speeds up the development or fine-tuning of methods. Of course, if you're buying a ready-to-go method, this is not so important, but there's always room for adding extra workflows into your day-to-day analysis. And we have some nice reflective injection logic going on in here. I'm actually not going to spend time on that today because we're looking at standardized runs for a particular application. So the early maintenance feedback, really, as I sort of said before, it keeps the user and the lab productive for longer, and it keeps the instrument at peak performance. So we get this kind of advanced feedback that is predictive rather than reactive. It's a bit like a battery bar. So you see the panels or the bars moving across, and therefore when you're approaching the time when you need to perform a maintenance event, it's already there. You can schedule it in. And the other nice thing is you can also, if you have an adverse event, you can detect and pinpoint exactly when and where that event has occurred. One of the defining features, I would say, that we have available on all our triple quads is the back shield. And this was designed really to maximize instrument uptime and improve the ease of maintenance by allowing the user to quickly and easily remove the ion injector capillary from routine cleaning cycles. So typically, in older instruments, you would have to vent the instrument, remove that ion injector capillary, switch it over, and then pump it down. So you'd probably schedule to do that overnight. But now what you can do is, it's really nice. You have two capillaries. You simply pull one out, put the new one in, and then within 30 minutes maximum, you're back up and running. You can then clean the capillary you've just removed at your leisure when you have time. I mentioned about the method optimizer earlier, and how to, when you don't have a ready-to-use method, such as the targeted metabolomics, lipidomics, or in this case, one of the biocrates kits, and you need to then optimize your entire method, both in the source parameters for each individual MRM. And if you are using hundreds of metabolites or transitions, then obviously doing this manually is not possible. You can take a method, a chromatographic method with the compounds that you've already got. If you've got knowledge of what the fragments should be, you can put that in, or you can ask the software to find them for you. And you can choose exactly all the things that you want to optimize within your run. So it's a really nice, useful thing. The other thing which I think is a really great feature is the ion optic rail has the ability to reduce the quadrupole isolation window. And this really increases specificity of certain MRM transitions. It's not always necessary, but since the quadrupole resolution is controlled on a per MRM basis, we can now improve our selectivity by implementing a narrow unit resolution rather than a unit. And you can see in these examples that chemical noise can actually be reduced once you narrow that window. So the specificity of the ion precursor ion going through is much better. And this is a really nice example, although it's not a compound that's relevant for today, but it's a really nice example of showing where actually narrowing down that precursor selection, we can now see this PFAS peak that was previously hidden in the background. Another critical feature for running these discovery workshows is that both pos and neg polarities are required. When you're running hundreds of MRMs, you must maximize that duty cycle. So you want to be able to achieve both the best signal, but also the number of points across your peak, because you need your method to be quantitative as well. And so the Agilent solution to this is to actually group the MRMs by polarity. So in each scan, you will actually, in fact, only get one polarity switching cycle. And the other clever thing that the software does is it now orders these in mass. And so it scans from high to low, because that is the fastest way of doing it. So here, you're really minimizing your switching time. And so it doesn't impact negatively onto your samples. And also, if you've got fast gradients and low dwell times, or if, as we'll probably see later, flow injection analysis where you've got no chromatography at all, you will need low dwell time. So you need that ability to really switch between the polarities very quickly, or between MRMs, very quickly. So when you have chromatography, one of the nice things to do is to implement a dynamic MRM approach. So we can see here that if we just do time segments, within that time segment, we're cycling through all of the compounds that come out in this example between 5 and 6 minutes. If we do a dynamic MRM approach, we make use of that retention time, but then we can also build in a tolerance window. So now all of a sudden, there's only a very small portion of that time segment where you're concurrently cycling through all of those compounds. And so what it actually looks like is this: when you input all of your transitions with your retention times and your retention time tolerance window, the software will now optimize the dwell times for all of your transitions. So if you, for example, in this example, you've got 123 concurrent transitions, it will now maximize those so that you have this dwell time of 1.55 milliseconds in this particular example. And it's a really nice visual. So then when we come to look at our data and we extract all of those MRMs, so in this example, we've got over 1,000 MRMs. You can see that we've got fantastic peak shape and signal. And the nice thing about this is if we go back to the sort of our discovery workflow, we've now got an end-to-end approach in that sort of now what we would call maybe pseudo-discovery, because it's a targeted method, but it's covering a whole panel of metabolites. And this example just shows how you can then do chemometrics. There were 5 cell matrix samples that were spiked with different levels of metabolites. As a proof of principle, you could then do a principal components analysis, and we can now see that each of the groups of cells are now clustering together really nicely. And then when we do some hierarchical clustering, we can now monitor the trends across each of those different groups of samples. It just so happens that in this example, it's concentration, but it could be treatments or strains of something, however you want to design your experiment. So hopefully you will see how sort of an intelligent mass spectrometer can be backed with a highly curated workflow. It can decrease your method development time and then accelerate your research. Now within Agilent, as I said, we have these targeted metabolomics and lipidomics workflows. But what is really nice is that the biocrates kit actually sits and complements across both of these workflows. And so you've got, it's fully optimized, standardized, it's quantifiable, and of course, fully supported by biocrates. So run your complete panel for a pseudo-discovery approach, understand the changes and differences happening between your samples, and then select your newly identified key metabolites to develop a routine targeted assay. Thank you very much for listening.